All right. Good morning. Do I have a microphone?
Yes, you do.
Oh, good. Okay. Welcome, everyone. I'm really pleased to have everybody back for our annual alumni meeting. This is a day we look forward to every year. It's very nice to welcome everyone back and also to share some interesting cases and some interesting presentations. This year, our format will be similar to many previous years. We have a couple of talks this morning by some faculty members. Then we will introduce and present our award to Dr. Robin Young, who's well known to all of us and has been a friend and colleague to many of us for many years. We'll have a break for lunch, and then we'll enjoy an afternoon of case presentations followed by dinner. We're back at the Water Club this year. Some of you may remember that the barge that it's located on broke loose during the hurricane a few years back, and we lost access to that venue, but we're back there, and it should be a very nice evening starting at 6 o'clock. Before I introduce our first speaker, I just wanted to make a couple of quick announcements. For those of you who haven't been back to visit the department for a few years, I don't think you'd recognize it. We completed a 25,000 square foot expansion in the last year, and it's largely occupied at this point. So I invite you, perhaps during lunch or a break, to wander over and see the new facilities. It's a very nice, and we all have all the attendings are back in the central area now, which is nice. The other thing I wanted to mention is last year we conferred the first of a new award, the William L. Gerald Award, for up-and-coming research in pathology. And after some discussion, we decided to separate that to a distinct event, which I'd be happy to invite all of you to join. It will be on March 31st, a few weeks after the USCAP meeting, and it will be a pathology research day to honor Dr. Gerald, to allow the pathology group to present some of their ongoing research, and then to confer the award on Dr. David Huntsman. So we will be emailing you more information about that in the spring. All right, so without further commentary, I would like to introduce our first speaker. And actually, it's not a speaker, it's two speakers. Dr. Robert Soslow and Dr. Britta Weigelt. Rob, as you probably know, is the director of GYN pathology and the director of translational research in our department. And Britta is a member of our experimental pathology division working in gynecologic cancers and breast cancer. And they've decided to do a tag team presentation today entitled Expanding Upon the Morphologic Characterization of Gynecologic Neoplasms, a Geneticist's Guide to Discovery. And this is going to be a unique format, so I'm introducing them together, and they're going to present together. Thank you very much.
Good morning, everyone, and welcome to Pathology Alumni Day. It's wonderful to see a lot of familiar faces in the audience. And thank you, David, for that very nice introduction. And Dr. Young, we're here to celebrate your magnificent contributions to our work. And we're very proud and very honored to have you amongst us today. And we hope that Britta's and my presentation will do you justice. At least that's the goal. Dr. Young is probably best known as one of the most thorough and careful morphologists of his generation, or perhaps any generation. And one of the themes that we would like to convey today is that the detailed review of glass slides and morphologic interpretation is as relevant now as it ever has been. And in order to tell the story, Britta and I have chosen to discuss two types of tumors that are near and dear to the hearts of not only Dr. Young, but also his mentor, Dr. Scully. And to emphasize the value of careful clinical annotation and morphological evaluation, what we're going to do is show you the way that Dr. Young's work has provided basically the groundwork for a geneticist to teach us more and more about these very special entities that he described. This is a summary of the talk. Britta will be talking about massively parallel sequencing. I'll talk about special histologic types of cancers. Again, there are two of them that are near and dear to Dr. Young's heart. We'll discuss disease-defining genetic alterations and spend even more time on those two neoplasms. I'm going to hand the microphone over now to Britta Weigelt, and I'll be back in just a minute.
Thank you so much, Rob, and good morning, everybody. Thanks for being here. So I will give a brief overview of the large-scale massively parallel sequencing analyses that have been performed over the past years and what we have learned from it so far. And I will mainly focus on the work by the Cancer Genome Atlas, or short TCGA, but there are also other consortia such as ICGC and, of course, also individual investigators that have performed this type of analysis. So the TCGA focuses on the omics characterization of 20 common cancer types to define the alterations at the DNA level, but also at the RNA, the epigenetic, and the protein levels. But in particular, the characterization of the somatic mutations using massively parallel sequencing really has revolutionized in the last years how we perceive cancer genetics. And novel findings have been made. And I think I summed up here 4 of them. I think the emergence of novel subtypes of cancer based on genomics data have been very important. For example, the POL-E subtype in endometrial cancer, you may have heard about it yesterday. So this is a subtype of tumors that is ultra-mutated, has many mutations due to a mutation in the polymerase epsilon gene, but is associated with a good outcome. Then novel targetable mutations have been identified due to sequencing, which may change therapy approaches that have been used so far. But I think we have also come to terms with the fact that common types of cancer are much more heterogeneous at the genetic level than we initially thought. So we now have learned that there's only a small number of highly recurrent mutations or highly recurrently mutated genes, and that there's quite a large number of mutations that affect only very few cases. And finally, new categories of cancer genes have been identified through these sequencing exercises. And these are genes which in the past have not even perceived to play any role really in cancer, but now have been found to be commonly mutated and even to be likely causative of cancer. And I'm going to introduce 2 of these new classes of cancer genes to you. The first one are the chromatin remodeling genes, in particular the SWI/SNF complex. So this is a large multi-unit protein complex that regulates nucleosomes and therewith transcription. And, you can use the mouse here. So as you see here by the red stars, there have been several of these members have been found to be targeted by mutations in a variety of tumors at a surprisingly high frequency, and it's now thought that about 20% of cancers harbor mutations in these chromatin remodeling genes. The second class I want to introduce here are the RNA machinery genes. So it has been found that there are several components in the microRNA biogenesis machinery, so RNA interference is shown here on the left side of the slide, that are altered in cancer, and also mutations in genes that play a role in RNA splicing, shown here on the right side, have been found to be mutated again in a variety of cancer types, and these mutations lead to a deregulation of gene expression and ultimately protein production. So it has become also clear now that there is a genetic basis for some of the seminal histologic observations that have been made by some brilliant pathologists, and that there are genotypic-phenotypic correlations in cancer. And as I mentioned before, and shown here on the left side of this slide, the common types of cancer are not only diverse at the phenotypic level, but also have been found to be genetically heterogeneous, whereas there are some rare types of cancer shown here on the right, which are rather homogeneous, both morphologically as well as at the genetic level. And these rare types of cancer are much more likely to be underpinned either by a specific constellation of mutations or genetic alterations, or even pathognomonic mutations. And by combining pathology with genomics, it has been shown that new cancer genes or even pathognomonic genetic alterations can be identified. And I give you 3 examples here. So on the left, Göran Stenman's lab showed that adenoid cystic carcinomas of the salivary gland and the breast are characterized by the presence of the MYB-NFIB fusion gene. Dr. Hansman's lab showed that granulosa cell tumors of the ovary are underpinned by mutations targeting the FOXL2 gene. And our lab found recently that the polymorphous low-grade adenocarcinomas are characterized by mutations in PRKD1. And the interesting part is working with these rare malignancies, you don't need to use this brute force approach like TCGA where you sequence hundreds and hundreds of cases, but usually just a handful of cases is sufficient. So I looked it up. There were only 4 cases of granulosa cell tumors that were sequenced to find FOXL2 mutations, and we actually went overboard with our 6 cases of polymorphous low-grade adenocarcinomas to find these new disease-defining genetic alterations. So, but coming back to Dr. Young, we think that Dr. Young's work is a great example of how one can bring the different pieces of the puzzle together, even when the technologies to solve the puzzle were not really available yet. But by combining the special histologic types of cancer with careful histopathology and also the observations of family history, really provided a basis for us geneticists and a basis for the identification of new cancer genes. And with this, I'm going to hand back to Dr. Soslow.
So let me just backtrack once to show you here. We have a small cell hypercalcemic carcinoma here, underpinned by a mutation in BRG1. And then we have an intermediate differentiation Sertoli-Leydig cell tumor, which, as we're about to tell you, is underpinned in many, but not all cases, by mutations in DICER1. First, a quick clinical and morphological summary of small cell carcinoma of hypercalcemic type. I consider this to be the seminal paper on the topic. Under the tutelage of Drs Scully and Young, this group studied over 150 cases of this very rare tumor. This is a brief summary of the tumor type and some of the clinical manifestations. As you probably know, these are highly aggressive tumors that tend to arise in women in the 2nd or 3rd decade, and the ovaries that are involved tend to be unilaterally enlarged. At least 50% of patients have extraovarian spread at the time of diagnosis. Nearly 2/3 have hypercalcemia. And despite the fact that we call this the small cell carcinoma of hypercalcemic type, there's also a large cell variant. And actually, in many cases, we find combinations of both small and large cells. And again, despite this nomenclature, small cell carcinoma, it's important to emphasize that this is not a neuroendocrine carcinoma. And because of its appearance and also because of the age group in which this tumor frequently presents, there is a wide and actually very difficult differential diagnosis. So here is a brief morphologic tour of small cell hypercalcemic carcinoma. In this field, you see a diffuse proliferation of small uniform cells with scant cytoplasm. You get the feeling that there's some dishesion here. And as we look through this tumor, we can find further evidence of this kind of dishesion. And when it becomes very well developed, we have the formation of these pseudofollicles that look rather like the follicles of juvenile granulosa cell tumor. Here is the contrast between an area in a small cell hypercalcemic carcinoma composed of small cells and another area composed of large cells. And in rare cases, we find only the large cell variant. And the large cell variant is characterized not only by the size of the cells, but also the rhabdoid appearance of many of them. This is the differential diagnosis for small cell hypercalcemic carcinoma. It's probably not even a complete differential diagnosis. I won't have time actually to talk about specifics regarding how we differentiate small cell hypercalcemic carcinoma from these other tumors, but suffice it to say that workers from Memorial Sloan Kettering and other institutions have come up with a potentially very powerful way of being able to confirm a diagnosis of small cell hypercalcemic carcinoma. So when I went back and read through the seminal paper published by Drs. Young, Oliva, and Scully, I was very interested to find these two sentences. I had actually remembered reading this when the paper came out, and I kind of filed it away back there somewhere. And it wasn't until recently that I realized how prescient, if you will, Dr. Young and Scully's work had been. So, first sentence is, 2 patients afflicted with small cell hypercalcemic carcinoma were a mother and daughter, and 2 were first cousins. And in another case, the patient's mother was reported to have died of ovarian cancer at 31 years of age, of course, suggesting that there might be a heritable component to small cell hypercalcemic carcinoma. And in fact, in late 2013, we have what I believe is probably the first paper describing the presence of germline BRG1 mutations in a family with hypercalcemic small cell carcinoma in Poland. I am now going to turn the microphone over to Britta, and she's going to tell you a little bit more about the SMARCA4 gene and its relevance to small cell hypercalcemic carcinoma.
Thank you, Rob. So SMARCA4 has not only been found to be mutated in the germline of familial cases of small cell carcinoma of hypercalcemic type, but last year there were 3 studies, one also here from Memorial, but Doug Levine's team, that showed that also sporadic small cell carcinoma of hypercalcemic type are characterized by somatic mutations in SMARCA4. And in these studies, about 75 to 100% of sporadic cases were found to harbor inactivating somatic mutations of SMARCA4. And there are, of course, other cancer types that harbor mutations in SMARCA4, as shown here in the bottom figure, but the type of mutations are different, as shown by the color coding. Whereas in the ovarian small cell tumors, you find non-missense mutations, meaning truncating mutations. The vast majority of SMARCA4 mutations in other cancer types are missense mutations, shown here in blue, such as stomach, head and neck, or melanomas. What is SMARCA4? So SMARCA4 is a tumor suppressor and a member of the SWE/SNF chromatin remodeling complex I introduced to you earlier. Again, it remodels nucleosomes and regulates transcription, and as Rob already mentioned, the protein product of SMARCA4 is called BRG1. So how could one use this information if one wanted to diagnose small cell carcinomas of hypercalcemic type? One could, of course, use straightforward sequencing of the SMARCA4 gene, but in these studies published last year, it has also been shown that these inactivating mutations lead to loss of the protein product BRG1, and that one could probably use immunohistochemistry to identify these cases. And there have been 2 recent follow-up studies, large studies, to define the specificity of the SMARCA4 or BRG1 loss in ovarian and uterine cancers. And one of these studies here on the right was actually by Dr. Soslow's team. So what they showed is first to confirm that indeed more than 90% of small cell carcinomas of hyperglycemic type show loss of expression of BRG1 as defined by immunohistochemistry. But interestingly, most of the other ovarian and uterine tumors that were studied, again by immunohistochemistry, retained expression of SMARCA4 and BRG1, if you want, with very few exceptions. So there are about 3 to 4% of ovarian clear cell carcinomas that show loss of BRG1 expression, and a subset of endometrial stromal sarcomas also are negative for BRG1. And there was also one interesting non-gynecologic tumor that was identified to be BRG1 negative, and Rob is going to tell you more about this.
Thanks, Britta. So this is really just going to summarize our paper that's in press in American Journal of Surgical Pathology, and I'm not sure whether you were aware, Dr. Young, but a number of these cases that we studied did come from Mass General Hospital. Are you a co-author on this paper? I think you may even be a co-author on this paper. So here is a typical example of a small cell hypercalcemic carcinoma, immunohistochemically stained with an antibody against BRG1, and we see retained staining in the non-neoplastic stromal cells, but loss of staining in the nuclei of the tumor cells. Here's a higher power shot of the same case showing these very nice follicle-like spaces, and another example in which the tumor cell nuclei have lost expression of BRG1. We studied a large number of other ovarian tumor types and a selected number of metastatic tumors to the ovary, and found with only very rare exceptions that these tumors, particularly the tumors that feature in the differential diagnosis with small cell hypercalcemic carcinoma, retain expression of BRG1. This is an adult granulosa cell tumor. This is a juvenile granulosa cell tumor. This is a Sertoli cell tumor, and I believe this is part of a Sertoli-Leydig cell tumor. There are occasional exceptions, however. As Britta mentioned, there was one clear cell carcinoma that did show loss of expression in tumor cell nuclei with retention of staining in the non-neoplastic stroma, but in general, clear cell carcinomas retain expression of this marker. The other exception that we came across in our study was in a case of metastatic melanoma to the ovary, which in some cases can form follicle-like spaces and therefore features in the differential diagnosis with small cell hypercalcemic carcinoma. And in one of, I think we had 5 or 6 cases, there was a loss of expression of BRG1, which had been previously reported in the context of melanoma, but still the vast majority, based on literature review, do retain expression of this marker. So the summary for the first part of the presentation is that germline SMARCA4 mutations are found in familial cases of small cell hypercalcemic carcinoma, but that even sporadic examples of small cell hypercalcemic carcinoma do show mutations in the SMARCA4 gene, and these would be somatic mutations. We and others have found that the loss of expression of the BRG1 protein is virtually diagnostic of small cell hypercalcemic carcinoma in the proper context, and perhaps that the restoration of the SWI/SNF chromatin remodeling complex might become a way of therapeutically targeting this complex and thereby benefiting the patients who otherwise have a really dismal prognosis. Let's now turn our attention to another tumor that I know Dr. Young loves, ovarian Sertoli-Leydig cell tumors, and again, he and Dr. Scully are responsible for publishing the most important seminal papers on the topic, and he was on a good run. We have one published here in 1985 where he and Dr. Scully reported on 207 cases, and then there were further publications highlighting specific variants of Sertoli-Leydig cell tumor, Sertoli-Leydig cell tumors with a retiform pattern, Sertoli-Leydig cell tumors with heterologous elements of the gastrointestinal type, and Sertoli-Leydig cell tumor with heterologous elements, including rhabdomyoblastic differentiation. When we think about Sertoli-Leydig cell tumors, you really need to keep 2 concepts in mind, and the first is that we essentially grade Sertoli-Leydig cell tumors primarily based upon the degree of Sertoli-like differentiation, so that all Sertoli-Leydig cell tumors can be segregated into one of these 3 categories, and there are some very interesting clinical pathological correlations that Drs. Young and Scully have taught us about. This is an example of a highly differentiated, this is the Sertoli-form area of a Sertoli-Leydig cell tumor, and the tubules are very well formed. So in the presence of Leydig cells, this would be diagnosed as a well-differentiated Sertoli-Leydig cell tumor. This is the typical low-power appearance of an intermediate differentiation Sertoli-Leydig cell tumor. Instead of finding a sea of well-differentiated Sertoli-form tubules, here we see nodules of blue cells with hypocellular areas interposed between the cellular nodules. And at higher power, what we can see here is that the tubule differentiation is much less well-developed. And seen here in many cases, the tubules can have a ribbon-like or trabecular appearance. And then we move into the poorly differentiated tumors. One important thing to remember about the poorly differentiated Sertoli-Leydig cell tumors is that for the most part, these have a sarcomatoid appearance. So one of the things that I teach the fellows is when you think you see a sarcoma in the ovary, particularly if the patient is in her 20s or 30s, the first thing that you think about is poorly differentiated Sertoli-Leydig cell tumor. In general, these tumors are overgrown by undifferentiated epithelioid to spindle cells with a very high mitotic rate and frequent necrosis. How do you recognize this as a Sertoli-Leydig cell tumor? You need to find better differentiated elements, which are frequently present at the periphery of these sarcomatoid areas. So the other thing to keep in mind as you go about analyzing the Sertoli-Leydig cell tumor under your scope is not only to decide what type of differentiation it shows, but also whether it shows retiform features and or heterologous elements. And here's a summary of the clinical pathological features of the retiform Sertoli-Leydig cell tumors and the variety of different types of heterologous elements that you can find in the context of Sertoli-Leydig cell tumor. Retiform SLT in particular can be a difficult diagnosis because when it is cystic, it may look a lot like a serous borderline tumor or a serous carcinoma. And with respect to the tumors with heterologous elements, it's really important here to distinguish the types that have rhabdomyoblastic, chondroid, and neuroectodermal elements from those that have gastrointestinal elements only. And that's because the tumors with rhabdomyoblastic differentiation in particular are very malignant, and this contrasts with essentially all of the other types of Sertoli-Leydig cell tumors that we see. So here's just a little tour through the morphology. This is a Sertoli-Leydig cell tumor with retiform Sertoliform tubules. Here's an example of a Sertoli-Leydig cell tumor showing gastrointestinal differentiation, which again, in the context of an SLT, probably does not have a lot of clinical significance. But when you find rhabdomyoblastic differentiation, it definitely puts the tumor in a very high-risk category. Again, going back through the seminal work of Drs. Young and Scully, there are some very interesting little tidbits. Here are two of them. In their review of these Sertoli-Leydig cell tumors, it turned out that I think of the 207 patients that had been studied had also had a sarcoma botryoides of the cervix, which I believed had been diagnosed before the emergence of the Sertoli-Leydig cell tumor. And interestingly, another patient had a multinodular goiter, and yet another had hypothyroidism, and yet another had a malignant thyroid tumor. Again, just as with small cell hypercalcemic carcinoma, underlining or at least giving us the suspicion that in some cases there is a heritable component to Sertoli-Leydig cell tumors. It's recently been found that there is a Dicer-1 syndrome. So a Dicer-1 syndrome patients have a germline mutation in Dicer-1, which leads in many cases to the development in childhood of pleuropulmonary blastoma, ovarian sex cord stromal tumors, but in particular Sertoli-Leydig cell tumor, botryoid embryonal rhabdomyosarcoma, and thyroid abnormalities, in addition to many of these other types of abnormalities, indicating that indeed there is a germline syndrome responsible for some cases of Sertoli-Leydig cell tumor, and Drs. Young and Scully provided the initial hint to that kind of inspired the description of the Dicer-1 syndrome. And I'm now going to hand over the microphone to Britta, and she's going to tell us about the somatic Dicer-1 mutations. Britta.
Thank you so much, Rob, and it's genetics for the last time. So about 3 years after the Dicer-1 syndrome was found, so the familial cases, or that they were underpinned by Dicer-1 germline mutations, David Hansmann's lab found that a subset of non-epithelial ovarian cancers, in particular Sertoli-Leydig cell tumor, were characterized by recurrent somatic mutations in DICER1. What's interesting is that the type of mutation is very different depending on whether it's germline or somatic. And I'm showing you here an overview of the DICER1 protein with the different protein domains and the different colors, and then here the exons on top. And I hope you can appreciate that here in the bottom of the gray dots, these are the inactivating germline mutations, so truncating mutations that are spread all along the gene. But if you look at the somatic mutations that have been found in DICER1, they are just focused on a few hotspots in the RNase 3b domain of the protein, and these are all missense mutations. So what is DICER1? Again, following the introduction, DICER1 is an endoribonuclease. It plays a pivotal role in the maturation of microRNAs. So these are small non-coding RNAs that regulate gene expression post-transcriptionally. And there have been now several studies that try to understand what exactly do these DICER1 somatic mutations do. And it has been suggested that these somatic mutations lead to a loss of the enzymatic RNase 3b enzymatic activity, and by this, a global reduction of very particular microRNAs, and this in turn again leads to a deregulation of genes that play a role in the proliferation but also differentiation in the ovary. Again, the question of how specific are these somatic DICER1 mutations, and there has been the original study here in 2012, but there were two recent additional studies which looked at that, the first one by Gauvin et al. And the other one by our team, again in collaboration with Dr. Young and Dr. Oliver. And we confirmed indeed that the majority of Sertoli-Leydig cell tumors do harbor somatic hotspot mutations in DICER1, but that there are also other lesions that harbor these mutations, such as juvenile granulosa cell tumor. We also find a subset of gynandroblastomas to be DICER1 mutant Sertoli cell tumor, even yolk sac tumors and mature teratomas, suggesting that the somatic DICER1 hotspot mutations can be found in a spectrum of ovarian sex-coiled stromal tumors, but in particular in Sertoli-Leydig cell tumor. And that was it from my part, and I'm going to hand over to Rob again to finalize this story.
I will point out one more thing here regarding the Sertoli cell tumors. What I thought was one of the most exciting conclusions that we came to is that these DICER1 mutations were only found in gynecologic Sertoli cell tumors. We had tested a large number of testicular Sertoli cell tumors provided by Victor Reuter and his team and found them uniformly to be negative for somatic DICER1 mutations, which is provocative. I'm not sure I know exactly why that is, but certainly provides an area for further research. So in summary, Britta and I hope that we've conveyed the following to you. That a familial DICER1 syndrome does exist, and it involves the development of, amongst other tumor types, ovarian sex-coiled stromal tumors, especially Sertoli-Leydig cell tumors, that the somatic DICER1 mutations are undoubtedly more common than these germline or familial DICER syndromes, and that these occur in a spectrum of ovarian sex-coiled stromal tumors and also in embryonal rhabdomyosarcomas and in selected cases of mixed epithelial stromal tumors that contain rhabdomyoblasts, right? And finally, that DICER1 and FOXL2 mutational testing may help distinguish some Sertoli-Leydig cell tumors and adult granulosa cell tumors from non-Sertoli-Leydig cell tumors. Before wrapping up, we need to flatter Dr. Young a bit more because he deserves it. So Dr. Young's work was really visionary in many ways. The unique morphological features that he described suggested the presence of genetic characteristics that separate the entities we discussed today from other tumors that were previously thought to be related. Dr. Scully's and Dr. Young's careful clinical annotation hinted at the presence of causative germline mutations. and that by understanding the nature of these germline mutations, we are able to inform our current understanding of the corresponding pathogenic somatic mutations. So in summary, the work of Dr. Young, particularly along with Dr. Scully, provided a geneticist's guide to discovery using the basic tools of a bright and energetic and motivated pathology, great morphologic interpretation, careful clinical annotation, and dedication and love for his work. So we hope we did you justice, Dr. Young. We very much appreciate you being here today. And to the audience, I hope you learned something, and thank you very much.
We have time for a few questions, if there are any. Well, I have a question, and I don't know if there's an answer for this or not, but one of the interesting correlates that we keep seeing with recurrent mutations that span different tumor types is the presence of certain lines of differentiation. And I think your Dicer story is very interesting because among the different entities on that list, pleural pulmonary blastoma, rhabdo, Sertoli-Leydig cell, all have rhabdomyoblastic differentiation. And there are several other similar observations among genetic aberrations that seem to correlate with morphology. And I'm just wondering if either of you have any thoughts about how we go about understanding how that mutation translates into a specific phenotype.
I'm sure you have a comment, but I'll make a quick one first. Okay. So I'm sure Britta has a really smart comment to make, but the only comment I'll make is that part of the reason why we undertook the Dicer 1 project in the first place was based on our hypothesis that we would find Dicer 1 mutations only in Sertoli-Leydig cell tumors with rhabdomyoblastic differentiation. And in fact, we did not find any correlations between the type of differentiation or the presence of heterologous elements with the finding of a Dicer 1 mutation, making the story even more complicated.
Yeah, and making it even more complicated, I think we also found we had a carcinosarcoma with rhabdomyoblastic differentiation and also some adenosarcomas actually with rhabdomyoblastic differentiation, and they all had Dicer 1 mutations again. So how are we going to go about it? I don't have an answer really yet, but I think that for some parts we really see a correlation between the type of differentiation and the underlying genetics. But I think the numbers we studied so far are probably too small to really make a strong case out of this yet. But if you have some cases, always welcome.
If anybody has primitive tumors with rhabdomyoblastic features, we should definitely
send them to Britta.
Yes.
Send them to Britta. We'd be thrilled to study them.
Yeah.